Thank you, and thank you for coming tonight. So, obviously, I'm going to give you a little bit about our process that we went through to discover the AOP and what are our next steps. So what we've chosen to do is a team approach. We have, as you can see, our team involves experts from throughout the world, from throughout different stakeholder perspectives, and also from different scientific disciplines. We have immunologists, we have chemists, in vivo and in vitro experts. And I think that was really useful for us to be able to have people coming from different perspectives to share their expertise. We started working on the project a couple of years ago as part of a breakout group within a larger workshop. And then we each worked on some parts of the AOP ourselves, writing the sort of literature review piece, and then coming together for teleconferences since then to pull it all together. And it is a project within the OECD's AOP program. So we're trying to integrate it into that process and keep to the guidance that has been promulgated by OECD. Just briefly, sensitization of the respiratory tract is often referred to as occupational asthma. You find patients with both acute and chronic symptoms of the upper and lower airway, including wheezing, bronchoconstriction, but those can also develop chronically into chronic inflammation, airway remodeling. Just as with skin sensitization, you have a sensitization and elicitation phase. And many people don't realize this, but you can actually sensitize the respiratory tract via dermal exposure. Our focus for the AOP was specifically low molecular weight organic compounds, excluding metals and some other respiratory sensitizers. And the motivation for putting together the AOP is that there is a real regulatory need for assays to detect respiratory sensitizers. There aren't any validated methods in vivo or in vitro currently. The mechanism for respiratory sensitization is not as well understood as for skin sensitization, and so we'd like to put that information together and be able to use it to support the development of test methods. Some of the initial issues that we came across when we were putting together the AOP include there's really quite a sparse literature when compared to skin sensitization. There's literature available for related pathways, but what we really wanted to do is to be careful not to use information that we weren't completely sure was specifically applicable to low molecular weight respiratory sensitizers. So in most cases, unless we were certain that it was related, we excluded information. For example, with skin sensitizers with protein allergy, which is, there's quite a bit of literature there, but we wanted to keep it, at least initially, separate. For us, we also had a lot of discussion around defining the adverse outcome, because you could have sensitization and then elicitation upon challenge. And what we decided to do, just as in skin sensitization, was looking at the regulatory context of how the information was going to be used. Sensitization is, in fact, the adverse outcome that we chose to focus on. And finally, of course, because you currently have an AOP for skin sensitization, and we wanted to define our elements of our AOP within the context of that and within the context of the guidance of trying to build these pathways of AOPs, really keep that, ended up being a challenge for us, basically. So here's an outline of the AOP. As you can see, it's very similar to the skin sensitization AOP. What we've tried to do is highlight differences between the two types of sensitization. And so with the molecular initiating event, you can see that most respiratory sensitizers prefer to bind to lysine residues on proteins. And so it's the same molecular initiating event, yet different. The rest of the key events through the pathway, cellular danger signals, dendritic cell activation, T cell activation, those are all also similar in general. However, what you find is that cells and systems exposed to respiratory sensitizers tend to release cytokines and other gene pathways that are related to a T helper 2 response. That's sort of an IgE-mediated response rather than the skin sensitization. And so we have in vitro and in vivo evidence for most of this. And again, what we find is that there are pieces of the pathway where you can discriminate between respiratory and skin sensitizers, but that there are other parts of the pathway that are almost the same. For example, dendritic cell migration. You don't appear to be able to use that as a way to discriminate between respiratory and skin sensitizers, because even though the dendritic cells themselves are different, they're still migrating to the lymph nodes in the same way. So we have this evidence. However, we don't understand the why. We don't understand what it is about respiratory sensitizers and their chemistry that lead dendritic cells to process those haptens differently and present them differently to the T cells. So moving to evaluating the AOP, we did that within the context of a modified Bradford-Hill criteria suggested in the OECD guidance. And you can grade your evidence as weak, moderate, strong. And what we found is, for biological plausibility, is this mechanistic relationship between the key events well established consistent with biological knowledge, and we found that that was mostly the case. In terms of the essentiality of key events, what is the evidence that one key event must lead to the next key event? Can you block the upstream key event and avoid the happening of the next key event? And that is the case. Very limited information on that in the literature, however, limited to 1 or 2 hallmark sensitizers, but not a large breadth of information to be able to make that judgment. And again, for empirical support of the key events in terms of, in general, what is the preponderance of evidence is supportive. There's not a lot of inconsistency in the literature about how the pathway actually happens, but again, there's not a lot of information on a large range of sensitizers. Most studies are using a couple of hallmarks, and temporality and quantitative information is really limited to a couple of studies. So we also identified in this process research gaps, and that was one of our main aims, was to be able to direct new research to further the field. And I just bring them up to say, I won't belabor them, to say that these gaps do not prevent us from actually using the AOP for regulatory purposes. So what you can try to begin to do is to place the assays that are available in the literature into context with the key events. And so similarly to the AOP for skin sensitization, we do have in vitro and in vivo assays available, but again, not a lot of chemicals, not many chemicals have been tested in each of these assays, and most of them have not reached the level of reliability that's needed for regulatory use. We also started thinking about combining the two AOPs and to think about the two AOPs as a mini network of pathways. So what you can then think about is that where you see divergent key events, for example, the molecular initiating event, key event 3A, as I'll call it, you can use those as breakpoints to start to develop test methods that can distinguish between skin and respiratory sensitizers. And I think it also probably lends itself to thinking about a combined testing strategy so that if you have novel chemicals, you can use this information together to classify them as non-sensitizer, skin sensitizer, respiratory sensitizer. And what we find, actually, there's just a paper published last week with a dendritic cell line discriminating using gene expression pathways between those two types of sensitizers. So we are working this up into, we had a poster today. I can send you a copy if you want. Feel free to send me an email with some more information. And we're working this up for the literature, and it's beginning to put it into the wiki, and we'll be doing that over the summer. And then after that's done and we start to get some consensus around the AOP, we'll aim to be continuing to work through the OECD process to try to develop some of the available assays for regulatory use. So thanks very much for your attention, and let me know if you have any questions.